Now, so who here has been involved or is involved in clinical research, subject research? Handshake, maybe?
In a lab that did some human research, but I wasn't involved in any of the stuff.
Were they identifiable samples?
I don't know.
Okay. So we're going to go over that. And I'm going to warn you, you might have some trepidation about being involved in clinical research. There is a highly regulated environment, mostly due to our own misdeeds in the past. And every time anyone revises the rules to try and make it better, they tend to make it more complicated and another hoop we have to jump through. So just to remind everyone, it's okay to be a little cautious when entering the realm of clinical research, but there are people here, including myself, that can try to help you as much as we can, help guide you to make important decisions that will hopefully keep you out of FDA jail or other jails. So the first question is it research? So most of the regulations that govern research do not govern activities like quality assurance testing or educational testing. So the first question that's when someone comes to me and says, do I need IRB approval? It's first, is it research? And second, is it human subject research? So for it to be research, you have to, your intent has to be, you're going to set out systematically to collect data to answer a question. If you're doing that and it becomes research, are human subjects involved? They don't have to be people in front of you. If you are using someone's data that's identifiable, it's human subject research as well. So if you get a data set from Cerner in the hospital or the MFA EPIC system, and it has subject identifiers, and there's 19 of them that are called out, it is human subject research. You need IRB approval or their approval to conduct the research without identifiers. So just to let you know that. And interrupt me if you have any questions along the way.
So, Richard, maybe you already mentioned, what about de-identified?
De-identified data is not human subject research. My advice to anyone, though, is submit it to the IRB and let them in writing, see on the next slide, tell you it is not human subject research. That way, if anyone ever questions it, you pull out that letter, you say, I did my due diligence. The IRB said it's not human subject research.
That's really good advice, everyone.
And so I always try, if you were to come to me and say, I need to do this, I need their date of birth. Well, that's a PHI element. And my question back to you is, do you need their age or their date of birth? Because if you're just using their age, it becomes not identifiable. Even though you know that in 2022, someone that was born 2023, they were born in 63, they'd be 60. You're not giving the month and the year, or the month and the day. So it becomes not a human subject research identifier. So as I said, in every attempt to make things easier and better, we tend to make things more difficult. One of these is we have a habit of creating TLAs, three-letter acronyms. To us, they're second nature, but to anyone trying to learn this, it's alphabet soup. It's confusing. We use the same initials in different order, like CRF versus CFR. I understand it's difficult, and if I ever throw out a three-letter acronym that you don't know what it means, just raise your hand and say you just did it, because I do it all the time. For the clinicians, if there are any clinicians in the room, there's a difference between practicing medicine and conducting research. So there are times when physicians would recommend doing something, the practice of medicine, that is outside of research. So you certainly can use a drug for an off-label use with the FDA. Whether or not you can get the insurance company to pay for it, I don't know. But you can practice medicine and do that. That is not research just to say, you know what, the best course of action for this patient is to give them this drug, whether or not the FDA has approved it for that indication. If, however, you're going to treat the next 30 patients with that same drug to see what the outcome is going to be in a larger group of patients, then you start going into the realm of you're going to start doing research. So I've had investigators come and talk to me and then decide, well, you know what, I'll just go ahead and practice medicine for the next 30 subjects and then go to the IRB and ask permission to use their data to write a paper. If called to the stand, I'm going to tell whoever that we had this conversation, you decided not to say that it's research. And then by your actions going afterwards and saying it's now research, you've kind of violated the Code of Federal Regulations. So just be careful. It goes to intent. You cannot stop people from practicing medicine, but if your intent is to do research, say it up front, be consistent, and seek the approvals that you need to. Also, very important, if it was never written down, it never happened. So there are many times when we go about, as we just talked about, well, I talked to someone at the IRB and they said this wasn't research. Unless you took meeting notes with the name of that person, it didn't happen. So that's why my advice is always to get whatever you do documented. Whatever you do with research subjects, they should be documented either in the clinic note in the electronic medical record or in a note that you keep as part of your study records. Okay. So part of our bad history of doing bad things to people in the name of science, you've all heard of Tuskegee. We have irradiated mental health patients in the state of New York more recently than that to see what the effects of ionizing radiation is on people. So finally, after enough of these had been discovered, the federal government enacted the National Research Act in 1974. And it set out that define what the ethical principles are in the conduct of clinical research and develop the regulations around that. So part of that, and this is the first time you're going to see 45 CFR Part 46, that was developed by the Department of Health and Human Services, well, Health, Education, and Welfare, as it used to be called. Now it's Health, Human Services. Back as a consequence of this National Research Act. It's been revised multiple times. Most recent, the biggest changes in like 20 years were done in 2018. All in the hope that things would be better. Well, in that specific, one of the specific requirements now in 2018 that was enacted is, how many people have ever seen an informed consent document? How long was the document that you read? Anyone else ever find a short one? No, they've gotten longer. 20 years ago, we could get away with 6 pages. Now we're up to about 20. 2018 required us to add a 1-page summary to the front of the consent. So now we're at 21 pages. They didn't get rid of any of the other requirements, and we just make them longer and longer. And now potential subjects have to read an extra page. And that summary really doesn't provide much detail because you've only got 1 page to do it. So, again, the goal of making things easier for subjects, that 1-page summary just made it longer. So, so after Health and Education and Welfare Department enacted these guidelines for research under their purview, other departments within the federal government decided that's a great idea, we'll come along. So 45 CFR Part 46 became known as the common rule. because now 17 different departments in the US government have said, this is our common rule for conducting clinical research. So the Code of Federal Regulations, there are a number that dictate the conduct of clinical research. 45 CSR Part 46 is the common rule, as I just said. Most IRBs function under those regulations. Now, 21 CFR 312, this, you're not going to get quizzed. It's my job to know these and advise you what they say. Governs the conduct of drug research. So if you're going to do any research on any compound that is going to change the pharmacology of the body or is going to claim to have an effect on a disease, it falls under 312. So I have filed an IND for baking soda because back at our previous institution where Ed and I were at, there's basic scientists that were claiming that if you can alkalize the microenvironment, it'll stop the spread of tumor metastases. So we were pumping subjects full of 24 capsules of baking soda. The hard part was finding a drug manufacturer that would manufacture it to the quality above what Arm & Hammer puts in the box so that we could say it's a drug and give it to humans. That was the hard part. If you're going to use a device in investigational research, it falls under the rule of 812. Again, many of these, the text are similar, but there's some nuances between them. And finally, the last big regulation that was enacted is HIPAA. Do we all know what HIPAA is? Health Information Portability and Accountability Act. We can't go into someone's medical record without their permission. We can't use their data without permission. You can get HIPAA waivers, just like you can get consent waivers, but you have to file and get someone to tell you you don't need to get someone's permission. Now, one of the loopholes that you brought up is human subject research is related to living humans or their data. If someone has passed away, it becomes non-human subject research, and you don't need HIPAA authorization to do it. So there's many of research projects at my previous institution where I advise that there is enough subjects' data or patients' data that have expired. Do it on those subjects' data rather than trying to include live people in the data set, because once you do that, it becomes human subject research. Oh, I can just say, so the pendulum has swung multiple ways with HIPAA. The initial guidance was for us to include the language into our consent. We did that. And then it became, you need to separate it out. So we had a separate HIPAA authorization document. Now the pendulum swung back. For most cases, it's still included, except for federal government, overseen by the NCI Central IRB, they want it separate. So depending upon what IRB has oversight on the clinical research project, you may have one document, you may have two. Yes.
So for the HIPAA, if the people have passed away, does that mean they previously gave permission to use their samples? And it's just like, doesn't matter if it's de-identified now? Or does it matter if they gave permission or not? When they were still alive?
So we certainly, so at my previous institution, we had a large, are you talking samples or data?
I guess,
Oh.
I guess you were talking about data, but I was thinking about samples. So yeah, either one.
In general, if they have passed away and there's tissue and pathology, it can be considered exempt research because it's not a human subject anymore. But they're going to want to know why that tissue is laying around. So we had a huge biobank where people gave permission to use their tissue for research. We just had to get separate IRB approval for each project. In that case, it was exempt research because we already had permission to use that tissue for research. In instances where they were not part of the biobank already, it becomes an easier approval because it's not human subject research. Again, I submit just about everything because I don't want to be called out by saying, well, we don't think it's, or we think it's human subject research. Prove to me that all these people have expired. Whereas if I tell it to the IRB and they tell me, you go ahead. But it is considered non-human subject research.
That's just interesting because like a cadaver, like, you know, someone who just died, you're not allowed to use their organs if they didn't say you could. So it's just an interesting like line.
Right. So, I mean, don't know that we ever instituted, there was, can't remember whether it was here or my previous institution. We had a protocol for people to donate their body to science that could be enacted. I mean, we had it IRB approved. We could get the family's consent. Just again, to cover all the bases so that no one's saying we're stealing from the dead. So again, we try to do everything up front possible. But like pathology tissues in the hospital, there are slides full or file drawers full of slides for people that have expired. You can use that. Again, I would recommend you tell the IRB what you're going to do, and it's only on patients that have expired, because that explains what the tissue is doing laying around. IRB is going to want to know, where is this tissue? Why do you have this tissue laying around? So one of the consequences of the National Research Act, they formed a committee. This committee came up with what's called the Belmont Report. The Belmont Report outlines the 3 basic principles of ethical conduct of research in the United States. So the three principles are respect, beneficence, and justice. So respect for persons. If the person is alive, ask their permission. Beneficence is to minimize the harm and maximize the benefit. And justice is to try and make sure that it's equal, that one group of people aren't having all the risks and the other group of people are getting all the benefits. Along with this, so still to this day, there are regulatory differences between Europe, ourselves, Japan, other industrialized countries that have regulatory bodies that oversee drug use and research. In an attempt to get everyone to play by the same rules, they created a committee called the International Committee on Harmonization, and they agreed in explicit detail on a set of guidelines on how to conduct research. In principle, it means if you do that research in Europe, you can use that data to apply for FDA approval in the United States or the Japanese authority in Japan. In practice, it becomes a little bit more difficult because of some of the differences in ethnic populations. And so many times the FDA will want to see the percentage of Anglo versus Japanese subjects in research done in Japan. So depending upon the number of representative people in the research that you conducted, you may have to repeat some of the studies in these different territories in the world. However, these guidelines are considered what's called good clinical practice. Along with good clinical practice, there's also good manufacturing practice and good laboratory practice. So manufacturers that produce drugs have to follow GMP in order for their drugs to get approved in the United States. If you are doing a test to monitor for drug efficacy, you have to follow GLP. All of these, I have a book about that thick of GCP standards, and they go into explicit detail. In the United States, these are guidelines, not rules. We tend to follow them for the most part, but not the explicit crossing all the I's or all the T's and dotting the I's. The classic example I give you is if you're working with a European drug manufacturer, Sanofi. One of the GCP guidelines is that IRBs are organized and have adequate resources. I had to create a memo that our IRB is organized and adequately resourced. And the IRB sign it so I could send it to them to check that box that our IRB was adequately staffed and properly resourced and organized. So we don't do that for any US manufacturers. So it's an explicit set of details that's way more detailed than the FDA regulations. So just know that they're out there. If someone says, well, GCP states we should do this, I always like to ask them, show me where it says that. Because there are many things that have crept into the practice of research that aren't explicitly detailed in GCP.
Which I'm sorry, I missed it. What's the difference between GCP and GMP and GLP?
C is clinical. So good clinical practice, good manufacturing practice,
Okay.
laboratory practice. So GLP, the lab, the translational research lab at Moffitt, our old institution. GLP is every morning we calibrate every balance. We have to then QA it before we put any drug substance on that scale. We have to keep that record that all of those QA checks and QCs have been done. Every product that we weigh, you have to record the lot number, the expiration date. So it's traceability all through the steps in your lab. So it can be tedious.
An example, GMP would be like CAR T cells, right?
Our cell therapy facility will have to follow GMP. So again, everything's got to be traceable to the lots of the pipette tips, to when your pipette was last calibrated. All of that's got to be done. So just a quick overview of types of studies. I usually break them down into two: observational versus interventional, or whatever word you want to use. So interventional, you interact with the subject and change something. Change their behavior, give them a supplement, give them a different drug, use a device, and see how the disease or that person reacts. Observational studies is you still might interact with the subject, but you don't perturb the system. If we're going to take the next 100 patients that undergo chemotherapy for breast cancer patients and just monitor their outcome or their weight loss, that's an observational study. Even if they're getting experimental drug, they're following their normal course of activities. Now, if I say I'm going to throw a new drug in there, then it becomes more interventional. Just phases, there are people that are really hung up on correctly labeling the phase of clinical research studies. In fact, some of the people on our IRB get into arguments about whether this is phase 2 or phase 3. You can call a pig a duck. It doesn't make it a duck. Basically, phase 1 is the initial early stages of clinical research with a new compound or device. You're testing for side effects. You're not necessarily looking for an outcome of benefit versus no benefit. Phase 2 studies are larger. You're trying to narrow down the subjects to have a specific disease because you're trying to narrow in on whether this drug is going to have any efficacy or not in order to go to larger studies that are conducted in a phase 3, where you might have hundreds of people getting treatment plus experimental disease versus just regular treatment. It's not uncommon, 300 to 1,000 people in some of these studies, depending upon how big of a difference you're trying to detect and what your outcome is going to be.
Richard, if you're gonna repurpose a drug, say, it's already know what the adverse effects are for that drug. You still have to undergo a phase 1, or can you just skip that and go directly to phase 2?
You can skip that to go to phase 2. It really depends upon what you know about the drug. I mean, if it's been FDA approved to market for 10 years, you know, we give 100 milligrams of this once a day. And if your preclinical or other experiments say 100 milligrams is enough to give you what you need as far as a pharmacodynamic effect, if you don't want to test whether 150 is better than 100 or 50 still gets you the benefit you need, there's no reason to go to phase 1. The issue is going to be you complete the phase 2 and you didn't get the results that you thought. Is it because you didn't give them high enough dose? Which is always, in cancer, up till recently, more is always better until you are almost killing the patient. Then you know you've gone too far. So you don't have to follow these steps. And that's one of the things that has led me to not get hung up on whether this is a 2 or a 3 just because it's 200 patients. Well, is it a 2 or is it a 3? Don't care. What's the hypothesis? What's the objective? And can you meet that with the patient population you've identified? So, like I said, it used to be you had to do phase 1. It had to be labeled phase 1, then 2, then 3, and then people started creating 1/2 studies. Because the process of opening a study here is 4 to 6 months. At some places, it's 6 to 12 months. And so to bypass that, if you can combine them into one study and say, first part, we identify the dose and we're going to treat 20 people, you get rid of all of that lag time in between your phase 1 study and your phase 2. We have a study that's open right now that's a 2/3. So I'm not hung up on labels anymore.
Does care.
Certain very strict people. Again, the FDA is going to look at it and say, does the data support the indication you're trying to get approved? Yes. A study that's pretty large is probably going to be labeled as a phase 3. But if it was a phase 2/3, the FDA is not going to care. The FDA has approved for temporary marketing drugs based off phase 1 studies, just because the response was so dramatic. And then you're required to do the larger testing to get to what is the proportion of patients that get thrombocytopenia or too low platelets. So they'll need all of the data, but whether it was conducted as part of a phase 2 study or a phase 3 study, they don't care. Our IRB members, a couple of them in my committee, care. And I keep arguing that that's not the purpose of an IRB.
So I have seen clinical trials where they have accelerated dose. So I suspect that they first started with a low dose and don't could see an outcome. The dose, and then they included more subjects, and sometimes they even mix like the previous subjects with the accelerated dose. So how does that work?
I could spend an hour talking about phase 1 dose escalation strategy. But briefly, so classically, they have used a dose escalation strategy called modified Fibonacci, where let's say you start with 50 milligrams as your first dose, do 100 and then 150, and then 200. As you go up, the percentage increase decreases. 50 to 100 is 100%. 100 to 150 is 66%. It starts going lower and lower down to 33% increments. So always treat 3 patients at per dosage tier until we start seeing severe dose limiting toxicities, and then we have to expand it to 6. Well, what we were finding is our preclinical drug models in mice, rats, or dogs did not predict the correct dose, what we thought. So classically, people were trying to get to 5 dose escalations. That should be your maximum tolerated dose to go to phase 2. When you start reaching 10, you've now treated 30 people with suboptimal drug exposure. And these are patients with cancer. So you don't necessarily want to waste someone's participation on giving them a dose that's too low. So in the 90s, new paradigms were starting to be proposed. So one paradigm was 100% dose escalation instead of doing the modified Fibonacci where it gets lower until you start seeing toxicity, and then you would slow down. People took that and said, what if we treat 1 patient per dosage tier? So now 5 dosage tier is 5 people, not 15. So there are other mathematical ways to do this, Bayesian methods where you have to predetermine your probability of certain toxicities. People have used historical data to back estimate what the size of certain studies would have been using all of these different methods. There are some advantages to doing every way. It really depends on what drug and whether or not the maximum tolerated dose is really what we should be doing in here. Because as I said earlier, the paradigm has always been more is always better until you're about to kill the person. But that's with classical chemotherapy agents. Is it true with monoclonal antibodies? There's some data to suggest that it isn't. So all of this, every year there's at least another paper that goes back and looks at these different paradigms. In general, now I think we're to the point where everyone agrees with 100% dose escalations. Whether or not you're treating 1 or 3 people at every dose is a matter of comfort. Are you willing to potentially harm someone because you've made 100% dose escalation because the person that you picked for that previous dose that tolerated really well was really an outlier? And if had you treated 3 people, the other 2 people would have been sick. So there's advantages and disadvantages to everything. This is more of an art than a science. So, like I said, we can spend an hour talking about those escalations. Yes, ma'am.
Is this similar for like non-cancer studies, like for migraines or IBS? Do they do like a similar thing, or is it different because it's not cancer?
In some certain diseases, they do. In many certain diseases, they treat normal volunteers with the drug. So, migraine, you might take 20 college students and give it to them and see what side effects they have at whatever dose you're going to give. Because you're not going to necessarily harm them if it's not. So there are many studies that are done that way. Now, diabetic medication, you don't want to drop someone's blood sugar, right? So you usually do those studies in diabetic patients. So it depends upon the population and the drug, whether or not you potentially are going to harm them by giving them that drug. Phase 4 studies is just this general catch-all of studies required by the FDA to collect more safety data, because one of the not well-kept secrets is we don't do well collecting toxicity data or side effect data on FDA-approved drugs. There is a mechanism, there's a form on the FDA website where you as a patient or your doctor can fill out this form and alert the FDA that you had this bad reaction. Most people don't know about it. And so depending upon the number of subjects that were treated or patients treated before you submitted for FDA approval, they may require you to do a phase 4 study to collect that data in a structured manner so that the FDA is sure that they know that the side effect is 1% of the population that get it rather than 10%.
Actually, another question about this. So I know there's a lot of data collected about like symptoms or like side effects that people have. But it also, they could be experiencing something that's not cause. It might not be cause and effect. It might be something separate. So how do you like delineate what you think is actually a real side effect and what is just a coincidence of something else going on?
So part of that is by sheer numbers. So if it's a mild reaction and you've treated 1,000 people and it only happens in 2 out of those 1,000, it's probably serendipity. If it's severe, the sponsor of the drug is required to notify everyone conducting research with that drug. They then look in their database. So first of all, the physician that's caring for that patient/subject makes the delineation. I think this is possibly related because the day after I gave this person this drug, this happened. Now, depending upon the severity, you might want to give that drug to that patient again and see if it recurs. Obviously, if it's very severe, you wouldn't want to do that. So if that data is available, they'll report it to the study's drug sponsor. Drug sponsor then sends it out to all the other investigators to know that this potentially is a side effect. As part of that report, they say, in our database of treating 30 people, this has not been reported previously. So we leave it as probably related. So we will alert all future subjects by modifying the consent and say, this is a potential side effect. But it's only after you get into the hundreds of people that you have any sort of power to be able to say whether that was serendipity or not. It happens all the time. I mean, we have to report if a research subject gets hit by a bus because they didn't look to their left when they walked out the street. We don't think, unless they're a drug that causes drowsiness or something, that's caused by that study drug. It's just we have to report everything that happens to them. So it gets into the database so you can make adequate decisions as to whether or not this is really a side effect. And that's what happens in phase 4, because you're going to the FDA with lower numbers of subjects because your drug effect is very dramatic. FDA more recently has been willing to allow you to try and market the drug because of the potential benefit in a life-threatening disease. But they're going to require you to collect all of that data, and there have been drugs that have been taken off the market because in hindsight, either the toxicity was too bad in more subjects than we thought, or honestly, it didn't work at the same proportion once you start treating thousands of patients. So this is my study to scare you, and I chose lightning bolts. So the PI is ultimately responsible for everything that goes on. They're responsible for the welfare of the research subjects and anything the research staff does. There are plenty of people looking over their shoulder to make sure that they do the right thing. So obviously the IRB, this box. So the IRB will review reports. You have to submit a report every year telling the IRB what happens. If someone has a severe side effect, we have to report to them within 24 hours that it has happened. And they are free to do audits to make sure that we're following good clinical practice. The sponsor themselves will come in and look over our shoulders to make sure that we're following the protocol as written. The government, the FDA, can walk in the door any day they want. They know we're conducting these studies. They come any day they want. OHRP, the Office of Human Research Protections, is also another federal government agency that's in charge of overseeing the welfare of subjects. So they can show up any day, anytime they want. And the institution itself may have separate committees. So the Cancer Center, we have to create another committee called the Protocol Monitoring Committee, which is independent of the IRB, that oversees the conduct of the research study as well. Now, I talked briefly about this before, but IRB has separate categories for the risk of the subject or the risk of the study to the subject. Basically, any time you're going to give a drug to a person, it's going to have to go to full board. If it is minimal risk, it can go to an expedited review process. And that means it doesn't go necessarily to a full board review. There's two at GWIRB, there's 2 meetings a month. Expedited does not mean faster. It means a different process. So generally the chair and one other reviewer will review it and say, this meets the qualifications for expedited research. Please change these things. So if you respond back quickly, you can get quick expedited approval. If you don't respond quickly, it will be just as long as a full board review, or longer if you don't fix it. So I always try to get studies moved from full board down to an expedited review by advising faculty members and other people to use the identified data if they can, increase the intervention with the subject so it's no longer exceeds minimal risk. So I can take blood from someone, it's still minimal risk because in the course of their normal health care, they'll have blood samples drawn. As long as they don't exceed 550 mLs in every 8 weeks, it's still considered minimal risk to collect that amount of blood. So you can design your study to try and make it as minimal risk as possible and hope and keep it down so it becomes expeditable. So I put up some examples of things that could go through an expedited review process. So drugs that are commercially available, FDA approved and marketed. If I'm going to treat a breast cancer patient with Taxol, it's FDA approved for that indication, can be expedited review, depending upon what else I'm doing to that research subject. As I said, only blood samples, as long as you stay below the limit, it can be expedited. If I'm going to collect hair clippings or fingernail clippings or voided urine from people, that can be expedited as well because I'm not being invasive. Collection of data. So if I wanted to collect the weights of subjects over time as part of an observational study, it can be expedited as well because I'm not intervening with them. Exempt research are conditions that are spelled out in the federal regulations. There's 8 of them. I did not put them up here because, quite frankly, every time I need to refresh my memory and go back and reread the different categories. But generally, food type research, does this taste better than this coffee? That's exempt. Educational research, is it better to give a slide presentation versus me just handing it out to you? See who learns better. That could be considered exempt. So those type conditions, if you meet them, it becomes exempt. You still should file and let the IRB tell you it's exempt. So just kind of to wrap up, there are places at the university where they have, I can say, trying to help you. So the Office of Human Research runs the IRB. They also maintain the city training, the required training in order to participate in human subject research. Required to take 2 courses here, either biomedical or social and behavioral investigators training, and then HIPS training, which is HIPAA. I recommend you also do GCP. So we do that through a program called CITY, which is a collaboration of institutions of higher learning. It's maintained at the University of Miami. So that training is free. We also have Protocol Builder, which is a piece of software where I've loaded two two protocol templates, one for data studies and one for drug studies. That will help you write a protocol. I also have the templates in Word that I can give to you as well if you'd much rather work in Word. Protocol Builder is basically take Word and do it through SharePoint, where multiple people can edit different paragraphs at the same time. That's basically what protocol is. I have recommended text in both templates and in Protocol Builder or instructions. Please make sure you clearly write out your hypothesis as an instruction. There are other offices here that are also here to help you. I already talked about the IRB. The Office of Clinical Research is housed in the School of Medicine and Health Sciences. That is their website. They have some tools available and some guidance documents that unfortunately you need to be in the MFA to be able to open. We, of course, in the Cancer Center have a clinical trials office with a web page. And if you really wanted to contact the FDA, that's the website. I'm available to help. If any of you have any questions about clinical research or how to modify, create, or edit a clinical research protocol, that's my email address and that's my phone number. Feel free to contact me and I'll help you however I can. Does anyone have any other questions?
So what happens if someone dies during the clinical trial? Do you have to like halt it to make sure it wasn't from the treatment? How does that work?
You definitely have to review the circumstances around that death. Like I said, I give you the example. Someone gets hit by a bus. They die. They're on the clinical research study. But I explained that the person walked out in front of a bus. So I wouldn't have to hold. I have to report it to the FDA. If I'm the study sponsor, because I'm the principal investigator, I have to report it to the FDA within 5 working days. I have to report it to sponsor if it's Pfizer or whomever within 24 hours, then they have to notify the FDA. So all of these people would be asking, provide all this background information. If it is questionable whether or not the drug led to the death because it's an extension of a side effect. So if, back to diabetes, if someone dies from hypoglycemia, shock, and then passes away, the FDA may tell you to hold on for a little while we investigate this. But it really matters what the circumstances of the death are. If it's an exaggeration of a pharmacologic effect, you'll probably have to hold until you're sure that it's not going to happen.
So then what if it is? Do you just have to like completely stop the trial and then do you still I assume you still publish those results?
You could try to publish the results. It depends upon how many people you treated. If it happened on the second patient, it's going to be hard to publish. There are times when, again, if you're 40 patients into a phase 2 study out of 50 and you have a patient pass away, you can look to see, was there a drug interaction? It's one of the veins of my existence. Was there a drug interaction with that subject? Can you get any tissue or blood from the person that passed away to measure the drug concentration if you weren't doing it at that time? So it may not require you to drop the drug or stop the study. It's whether or not you can adequately explain why this reaction happened in this person, but not the other 39 people that you've treated. There are drugs that have been put back on the shelf because a bad thing has happened during studies. So again, it depends. If you've treated 500 people and a person passes away, it could be a bad reaction. That's 0.5%. If you can explain how you're going to either adequately monitor to make sure that someone, if it's something that progresses as the patient's treated over and over again with a drug, is it something that you can stop when 50% of that effect has happened?
Suppose that person dies because of a natural progression of the disease, like in cancer, then what are the ways through which you can prove that that's not because of the drug you gave, it's because of the progression of the disease? Or-
Usually there are other clinical symptoms of disease progression
Mhm.
that you can use to make that case. So, I mean, we know that people are progressing while we're treating them with a certain drug. They haven't yet met the criteria for progression that would kick them off the study yet. So if someone's tumors has to grow 30% bigger, if they've only gone to 20%, we have that scan, we know their tumor is getting worse. So if they happen to pass away, we know their tumor burden is increased. If they have clinical symptomatology that led to their demise, we can rationalize that it was due to drug or disease progression.
So is there any percentage limit to the number of persons who are who have participated in the disease and they passed away, by which I mean, is there any limit to so that you have to drop the drug, otherwise you can continue? Like you said, just 1 in 500 is too less. But other than that, is there any lower or the upper limit?
Well, it also depends upon the disease that you're studying. So our comfort level with bad side effects, treating someone with cancer because they're going to die, is a little higher than treating someone with a migraine that might walk around and get one migraine attack every couple of months. You know, that's one of the knocks against supplements that cause side effects, that it may prevent cancer, but people do not, healthy people don't want to walk around and have side effects of the drugs they take. So your threshold for what is what's acceptable is different based on the disease. Now, again, if 3 or 4 out of 10 people have died from progressive disease within a month of getting the experimental drug, your drug's not working. Unless you've got another 4 or 5 where their disease is melting away. So it's all in context as to what's acceptable toxicity and acceptable progression rate based on the disease of the person. Yes, ma'am.
So it's a little bit off topic. So say you're like in the middle of a phase 2 part of the trial where you have patients with the disease being tested with the treatment, and you have clear responders and non-responders, and you, since you're halfway through, you already have all the patients enrolled, and you want to have either a spin-off or like work into the study to see if there's some explanation for the response or the non-response. Does that require a new IRB? Do you have to like resubmit the whole thing to add that to a protocol, or is it like better to just have it as the de-identified stuff that's then being analyzed by a different lab, or how does that part work?
So if I had an idea that a certain lab test or research, I'm going to measure this weird protein that I think is a marker of response, I certainly can amend the study and say I'm going to start collecting them from everyone. So again, that can be a pretty rapid turnaround as far as IRBs can go. If I say, you know what, I noticed in 3 of the subjects that this protein X was increased and they responded, I didn't see it in any of the people that their disease progressed. The IRB might say, okay, draw an extra blood sample, draw an extra 2, 3 blood samples, as long as you don't go above this magical limit of 550 mLs in 8 weeks. You know, anything, again, the IRB is looking out for the safety of the subject, and adding an extra blood sample is minimal risk, and they would probably prove that quickly and with applause. But anytime you're going to deviate from the plan on purpose, you should get IRB approval ahead of time. Now, if a research subject went and got this test without my knowledge as PI, and they brought me that information, okay, I didn't deviate from the plan. The subject went and did this by themselves. If they got genetic testing, for instance, through Ancestry.com, and they brought me this data, I didn't tell them to do that. So But yeah, if you think you have a prognostic marker, I would add it while you treat the last couple of patients just to see. And then you can incorporate it formally with enough power in the next study to test whether it's statistically valid. Welcome. Yes, sir.
We have a undergraduate summer research program where we actually have all the students sign a consent form that they research subjects, and we you know, we follow them after the program where they end up applying to medical school or graduate school and all that. So I was surprised that you said educational studies like that are exempt, because we actually have an RFB protocol. And so are those subjective, you know, like the RFB, like if they're on a good day, there's, oh yeah, this is exam, and then on a bad day, there's a no, it's not.
Well, there's some of that, honestly, to what you just said. It also depends upon the way you describe it. So again, if it's a short interaction, I'm going to hand out my slides to this side of the room. I'm going to stand here and talk to the other side. That's a short-term outcome. I'm going to look at their tests and get 2 averages for how they perform. You're following people for the rest of their lives.
Well, not for the rest of their life, but for a while, yeah.
For a while. So I'm sure you want to gain their permission to do so.
I see. So that the duration of the study matters a lot. Okay.
The context. This is all about context. Yes, ma'am.
So my question is, when like an academic lab collaborates with, let's say, a pharma company to design drugs, and the drugs are tested in the clinical trials, how is the analysis and the design of the trial divided between the lab versus the company, like in terms of involvement, in terms of regulation, in terms of analysis or other experiments?
It can vary, and it depends upon the contractual arrangement between the lab. If you've licensed the drug that you discovered in your lab to Pfizer, Pfizer could say, thank you very much, we'll take it from here. Or they could say, we want you to continue to analyze blood samples or the marker of response or something. Now, if I was Pfizer, I would make sure that I'm sending you de-identified blood samples so that you're not participating in the research. You're doing stuff, but you're not doing human subject research. So that is clean, but they could also say, give me the procedure that you use for that assay, and we're going to send it to Covance or LabCorp, have them set it up. So it's an independent contractor that's doing this with no potential conflict of interest. Because depending upon the licensing agreement, your lab benefit from the drug working. So it can start one way and then go to the other. It's very common. Where we were from before, we had an independent translational research lab that would take an assay from Ed's lab, reproduce it, run it on the lab specimens that come from the research study. Because the guy's lab, we want to move on and do other research rather than contractually running these samples over and over again for 100 patients. So it depends upon the setup and the licensing agreement. But once you get into FDA regulated research, like if you enter phase 1, in order for that data to be presented to the FDA, it's got to be done in a GLP facility. So your lab is not GLP. So there is this cutoff where in phase 1, you can still do it. But once you want to really take that drug and you know you're going to submit data to the FDA, it's got to be in a GLP facility. Yes, ma'am.
Question, more regarding your opinion on this. So with the FDA no longer requiring animal research for some trials to move to human research, what is your opinion on how that will affect research in the future?
Not requiring it. So as I said really early on, sometimes we get the starting dose for phase 1 studies really wrong. Sometimes they take the animal data and there's a nomogram from converting the dose you gave to a rat to the dose equivalent in a human. Sometimes those nomograms aren't done well. Rodents have this wonderful capacity to metabolize drugs much faster than humans can. And so there have been times when we give a drug that we expect the half-life to be 4 hours and it turns out to be 24 hours. That's valuable information, except for all of those anecdotes where it doesn't work. My concern is we're going to end up picking a dose to start in humans that is wrong. And on one hand, starting with too low of a dose, you have participants that you theoretically have wasted by giving a drug that's too low of concentrations. But what's going to happen when you give someone too high of a dose as the first patient and they die? That's kind of scary. So I can tell you, if at Moffitt, where we were before, if we were the first time that drug had gone into a human being, had plenty of those studies, everyone was on edge. In fact, I was told to stop hovering. Sure that people still weren't turning purple, because it's kind of scary to give someone a compound that's never been given to a human being before. So I'm a little concerned about, I know our nomograms are okay, but they're not great. And if we're not going to base them on an animal, how are we picking that dose? So again, the pharmaceutical industry is very risk adverse. So I can see while that requirement for animal testing has been stated it's no longer required, I can foresee pharma keeping it just to try and have some support for what is the best first dose we're going to give to a human being to minimize bad things. Yes, ma'am.
Is there a specific like threshold of data that needs, like, I don't know if like that has to be or ends up being reported that then requires a phase 4 trial to be done on a particular drug, or is it just someone who is interested in doing it and has the patience? Like, does anyone ever say that enough has been collected that this needs to be done? I don't know if I'm asking this the right way.
I think I get the gist of what you're asking.
Yeah.
So in general, no one wants to do a phase 4 study. It costs money to do research. And so pharma sponsors will not conduct the phase 4 study unless they're told they have to by the FDA. Or it's for some other purpose. But, a phase 3 study, let me put it this way, a phase 2 study done here with 40 subjects is $1 million. If you're going to collect thousands of patients worth of data for phase 4, now the requirements are going to be less data being collected, but I'm not going to do it for free for Pfizer. They're going to have to pay us to collect that data because I have to hire people to do it. It can be very expensive. Now, a clinician who says, I want to understand the side effects of giving women with breast cancer Taxol, they can do that study. I guess by definition, it's a phase 4 study because it's FDA approved, it's post-marketing, but they're doing it for a reason. Create a registry, answer a question about the side effects, things like that. But a large phase 4 study that's mandated by the FDA costs some money. And well, I'll let you get on with dinner.
Thank you very much, Rich.
Thank you all for coming.
Just quickly, before we close,